New York Structural Biology Center, we opened our doors in 2002 in this facility, and the purpose was to provide very advanced instrumentation in structural biology to the 9 member institutions that had formed the consortium that is the New York Structural Biology Center. The institutions that make up the consortium are Columbia University, Rockefeller University, Memorial Sloan Kettering Cancer Center, Mount Sinai School of Medicine, Albert Einstein School of Medicine, Weill Cornell College of Medicine, the Wadsworth Center at the State Department of Health, City University of New York, and New York University.
The founding of the center was in fact a grand experiment. We had to bring together 9 biomedical institutions in the New York area. We've succeeded in doing that, and here we are a decade later, having established the center and the functions that it represents, the expertise it offers to our member institutions, and the opportunities it provides for the researchers to carry out their work. Going into the next decade, we are opening our instrumentation and expertise to the outside world, to the private sector, to other academic centers.
New York Structural Biology Center features 4 main facilities. These are Nuclear Magnetic Resonance, NMR Spectroscopy, Electron Microscopy, X-Ray Crystallography, and Protein Production. NMR Spectroscopy, they are supported by columns of concrete 30 feet high and about 16 feet in diameter that couple the machines down to Manhattan bedrock schist and thereby reduce the possible vibrations that are the enemy of these experiments.
In our facility, we have 3 solution 800 megahertz NMR spectrometers and 1 solid-state 750 megahertz spectrometer. One of the solution 800 megahertz spectrometers is equipped with an automated sample changer and a microprobe for screening libraries of small compounds against target proteins. Technology in structural biology advances quickly, and NYSBC strives to stay abreast of these changes. We have one of the first commercial dynamic nuclear polarization NMR spectrometers installed in the facility for applications to solid-state NMR, in which the technique provides up to a 100-fold increase in sensitivity. NYSBC has 2 900 megahertz spectrometers. Together with the other NMR spectrometers you've already seen, this makes the facility the largest facility in the United States for studies of biomacromolecules by NMR spectroscopy. This technique is complementary to the other technologies here in the center.
This microscope lets us reconstruct an entire cell or a group of cells at very high resolution. And so at this resolution we can see organelles, we can see mitochondria, we can see how the cells are interacting, we can see virus particles inside the cell, and we can see exactly what's going on. And this is a real breakthrough because this is a relatively new technology and it's only recently been applied to biological problems.
Our third capability at NYSBC in terms of instrumentation refers to X-ray crystallography. These instruments are used for some of our samples, but for the predominant set of things these days. And what we really need to do is go to a synchrotron source. The major source that we have is out on Long Island at Brookhaven National Laboratory. But that facility, the National Synchrotron Light Source, is being closed down and replaced by a new facility, NSLS2, which will be ready in 2015. This is called NYX. It's a micro-diffraction facility that will produce very fine, very bright, very parallel beams that will give us an unprecedented kind of capability. The heart of our protein production facility lies in robotic devices that we have developed to be able to clone and express proteins in a high-throughput manner, many things simultaneously. These capabilities are being used to investigate in an efficient manner many kinds of proteins, but in particular by the New York Consortium on Membrane Protein Structure, NICOMS, we are investigating membrane proteins, and that's to the benefit of many of our investigators, including Filippo Mancia from Columbia University.
My lab interacts very closely with the staff here at NYSBC. We do projects together, we do technology development together. And we've had fruitful collaborations involving exchange of data and information that have had two beneficial results. One is the optimization of the protein production and screening platform, which is successfully operational here for both prokaryotic and eukaryotic membrane proteins. The benefits have been tremendous, because I have been receiving and I've been working with membrane proteins that I receive, the clones of which I receive, I know will have a substantially increased probability of yielding a certain atomic resolution structure.
The future of the Structural Biology Center is bright. It will grow to include more participation from our member institutions, the outside users, and it's particularly important as structure becomes a greater part of biology across the spectrum. All those researchers will be looking to the Structural Biology Center for help and technology to solve their problems.